Hello, everyone. Thanks for joining today. My name is Natalie Delgado, and I'm a second-year pediatrics resident here at Duke. I'm going to be sharing with you all our project entitled Branched-Chain Amino Acid and Tryptophan Metabolism and the Pathogenesis of Youth-Onset Type 2 Diabetes Mellitus. As that name suggests, our focus was to understand the changes seen in amino acid metabolism, particularly of tryptophan in type 2 diabetic youth, and also to see if we can identify metabolomic signatures that differ significantly in the type 2 diabetic adolescents than non-diabetic. For some background, we have previously demonstrated that insulin resistance in youth is associated with elevated levels of branched-chain amino acids, or BCAAs. The BCAAs can compete with aromatic amino acids, including tryptophan, for uptake into the pancreatic beta cells. This is important because tryptophan is the precursor of serotonin, and serotonin has been shown to increase beta cell mass and glucose-dependent insulin secretion. We then hypothesized that elevated levels of BCAAs could induce diversion of tryptophan metabolism to production of kynurenine instead of serotonin. To test this, we analyzed 24-hour and spot urine samples and compared levels of byproducts of BCAAs and tryptophan metabolism in 3 different groups. Our first group was comprised of 56 non-diabetic adolescents with obesity. Our second group was 42 adolescents with a known diagnosis of type 2 diabetes mellitus. And our third group was 43 normal weight lean controls. We measured metabolites derived from branched-chain amino acid catabolism and tryptophan metabolism in spot and 24-hour urine samples using liquid chromatography and mass spectrometry. You can see in Table 1 the list of all the metabolites that we measured normalized to urine creatinine in the 3 different groups. What we found was that in 24-hour urine samples, levels of branched-chain keto acids, tryptophan, and kynurenine levels were higher in type 2 diabetes, significantly more so than in the other 2 groups. In addition, you'll see in Figure 1 and Figure 2, the level of kynurenine to tryptophan was higher in the type 2 diabetic group with the p-value of 0.011. In contrast to that, in Figure 2, the ratio of 5-HIAA, a marker of serotonin metabolism, to tryptophan was lower in the type 2 diabetic group. In addition to that, the ratio of 5-HIAA to kynurenine was also lower in the type 2 diabetic group. These results suggest that increased BCAAs are accompanied by diversion of tryptophan metabolism from the serotonin pathway to the kynurenine pathway, suggesting perturbations in amino acid metabolism in youth-onset type 2 diabetes. And these alterations could contribute to development of beta cell dysfunction and progression to type 2 diabetes in youth. In addition, the study shows that serotonin and its metabolites could serve as potential novel biomarkers in detecting progression from insulin resistance to type 2 diabetes, and that using spot and 24-hour urine metabolomic profiling could be a novel way to characterize metabolic status in adolescents. Thank you all for taking the time to listen today, and I am available to answer any questions.